Selectins, rolling leukocytes, and the inflammatory response. Human bodies are constantly exposed to a plethora of bacteria, viruses, and other inflammatory substances. To combat these infectious and toxic agents, the body has developed a carefully regulated inflammatory response system. Part of that response is the orderly migration of leukocytes to sites of inflammation. Leukocytes literally roll along the vascular wall and into the tissue site of inflammation. This rolling movement is mediated by reversible adhesive interactions between the leukocytes and the vascular surface. These interactions involve adhesion proteins called selectins, which are found both on the rolling leukocytes and on the endothelial cells of the vascular walls. Selectins have a characteristic domain structure consisting of an N-terminal extracellular lectin domain, a single epidermal growth factor domain, a series of 2 to 9 short consensus repeat domains, a single transmembrane segment, and a short cytoplasmic domain. Lectin domains first characterized in plants bind carbohydrates with high affinity and specificity. Selectins of 3 types are known: E-selectins, L-selectins, and P-selectins. L-selectin is found on the surface of leukocytes, including neutrophils and lymphocytes, and binds to carbohydrate ligands on endothelial cells. The presence of L-selectin is a necessary component of leukocyte rolling. P-selectin and E-selectin are located on the vascular endothelium and bind with carbohydrate ligands on leukocytes. Typical neutrophil cells possess 10,000 to 20,000 P-selectin binding sites. Selectins are exposed on the surfaces of their respective cells by exposure to inflammatory signal molecules such as histamine, hydrogen peroxide, and bacterial endotoxins. P-selectins, for example, are stored in intracellular granules and are transported to the cell membrane within seconds to minutes of exposure to a triggering agent. Substantial evidence supports the hypothesis that selectin carbohydrate ligand interactions modulate the rolling of leukocytes along the vascular wall. Studies with L-selectin deficient and P-selectin deficient leukocytes show that L-selectins mediate weaker adherence to the leukocyte to the vascular wall and promote faster rolling along the wall. P-selectins conversely promote stronger adherence and slower rolling. Thus, leukocyte rolling velocity in the inflammatory response can be modulated by variable exposure of P-selectins and L-selectins at the surface of the endothelial cells and leukocytes respectively. And that is selectins, rolling leukocytes, and the inflammatory response. Have a great day.